because of the war?
Yes, that is really something that's been hard, and yeah, it's been something that he's been.
Okay.
Hola, buenos días.
The signal that we are alive right now. Then let me introduce you, but I'm gonna do it in Spanish for the assistance, but after that you can have some information about this, okay?
Okay.
Hola, buenos días. Estamos aquí en el segundo día del foro, el 14º foro internacional de investigación y posgrado de la Facultad de Ciencias Naturales. Y es un placer para mí presentarles al doctor Michael Lefrano. Él es un investigador de la Universidad Politécnica de California. Él obtuvo su grado de doctorado, de su PhD en la Universidad de California en Davis, y tiene una amplia experiencia trabajando en el área de metabolomics, principalmente pues en problemas o temas relacionados con la nutrición. El día de hoy nos va a presentar un tema muy importante con relación a la vitamina A, y espero que para todos sea de gran ayuda esta información que nos presentará el día de hoy. Les recuerdo que ustedes pueden hacer sus preguntas a partir del botón verde que se activa en la plataforma, y si no, pues aquellos que tienen mi teléfono lo pueden también hacer a través de WhatsApp. Tenemos traducción simultánea y entonces, bueno, no espero que haya problemas en este aspecto para que ustedes entiendan mucho mejor este tema. El doctor Lefrano va a venir el próximo año a hacer una investigación, una estancia de investigación de 1 año aproximadamente aquí en la universidad, y esperamos, bueno, pues empezar muchas colaboraciones con él para el futuro. Bienvenido, welcome, Michael. If you want to start or introduce yourself, that's okay. Thank you.
All right. Thank you very much for the introduction. Before I start my presentation, I'd like to first thank you for inviting me. I'm happy to be here and talking about some of my recent work. Much of it has not been published yet, so there's a lot of new things that I'll be talking about. I have previously worked with Dr. Anaya Aloyola on some research, specifically some work that was done at the university, and I'm planning to visit the university at the beginning of next year as we will do some more research together. So I'm really looking forward to that, and also just seeing the great university that you have there.
Thank you.
So I will begin my conference right now.
Yes.
And today I'm going to be speaking about a global assessment of biomarkers of vitamin A deficiency and toxicity. And I say global assessment because we do have 4 studies that I'm going to be talking about that span across the world. They are smaller studies, but they do
Hay estudios más pequeños
a lot of different
que nosotros
areas.
podemos tener en muchas áreas distintas.
There's one that I'm going to first talk about that was done in the Philippines.
En las Filipinas.
Speak about one from Zambia.
Y podemos pensar de Zambia.
And another one from South Africa.
Y otro de
and lastly,
Sudáfrica,
a study
y por
from the
último,
USA.
un estudio de los Estados Unidos.
So global.
Can we stop a little bit, Michael?
Excuse me?
We are listening the Spanish version and the English version in the same session. I don't know if it's a technical problem, or we're gonna have this translation in the same session. Could anybody?
Okay, that's
¿Ahí
not a problem.
¿Ahí me escuchan?
Yes, sí, se escuchamos.
Pero ¿es en el mismo lugar, me estás diciendo?
Sí, estamos escuchando en inglés y en español en el mismo lugar.
A ver, dame 2 segundos, por favor.
Yeah. Sorry, Michael, just a few minutes.
Oh, it's no problem.
A ver ahí, 1, 2, 3, 1, 2, 3.
Ahí yo te escucho a ti, pero bueno, Michael ahorita no está hablando.
No, a ver, que hable, por favor. Dile tantito.
Michael, could you talk a little bit to see if we solve this
Absolutely,
problem?
yes. So I was going to be speaking
A ver, ahora voy a estar
vitamin A
hablando
deficiency
sobre
being a leading cause of morbidity and
las
mortality.
causas principales de morbimortalidad.
Thus,
Ahí
it's important to do
Seguimos
es-
oyendo las dos.
across
A ver, denme
the
5
span
minutos.
of
Díganle que por favor
different
se espere
countries.
5 segundos.
And
Michael, sorry for the
Yes.
interruption. Could you wait a little bit, please? 2 minutes.
absolutely, absolutely.
I'm sorry.
Sorry.
I'm very sorry.
Something
Oh, that's
has
okay.
happened.
No problem.
A ver. YouTube. ¿Qué onda? ¿Qué está pasando aquí?
Creo que ahí se oye más al fondo, no sé.
Oh, that's okay. No problem.
¿Crees que ya quedó?
A ver. Algo pasó porque la vez pasada estuvo perfecto, Dios mío. Déjenme salir de aquí. Y me voy a salir de aquí. Ay, Dios mío, qué horror. Qué desesperación. A ver ahí. Could we try again? ¿Podemos intentar de
Okay.
nuevo?
Okay, Michael, can we try again, please?
Yes. So I was going to be mentioning that children and pregnant
Y
women
voy
are
a estar mencionando a los niños y a las mujeres
about a
embarazadas.
third of preschool age children have vitamin A deficiency, about 19 million pregnant women. And there are some countries where it's highly prevalent, such as
Donde
in
es altamente
Africa
prevalente.
and Southeast Asia.
Ahora sí. En el
No,
sudeste de
seguimos
Asia.
oyéndote, pero
Ay, Dios
yo
mío.
creo que vamos a quitar la traducción o algo, porque
No, a ver,
ya...
voy a hacer algo diferente. Si quieren para no perder el tiempo, continuamos
Sí.
y déjame ver qué podemos.
Michael, you can continue, please.
Okay.
Okay.
So to give some background on vitamin A, it is a essential fat-soluble micronutrient. And there are multiple forms of vitamin A, and both those that have action in the body as well as different forms that are in the diet that we consume. And some are from animal sources such as retinol esters, whereas others that are in plants are called carotenoids. And some of these carotenoids are able to convert into vitamin A in the body. And the way that it primarily functions is that vitamin A in its retinoic acid form will bind to receptors in the nucleus and impact the transcription and translation of specific proteins that are involved in vision, immunity, organ development, reproduction. So a variety of different functions in the body. And these have been studied for many years, but there still are some things that we're still trying to learn about concerning vitamin A. And one is the effect of vitamin A on global metabolism. So there's still much to be learned about its effects on a variety of metabolites beyond some of the main things that we've focused on so far. And also looking for different biomarkers of vitamin A status, because there are some issues with the markers of vitamin A status that we have now today. And so metabolomics is a technique that might be useful for actually both of these purposes. So metabolomics is the large-scale analysis of metabolites. So in other words, substances formed in metabolism, and looking at those within biological fluids and tissues. So things such as blood and adipose. And it enables us to see metabolic changes in a broader context because we're looking at oftentimes hundreds to thousands of metabolites being changed in a single sample. And you can see here on this figure here how connected metabolism is. This represents all of the different intermediates and connects things such as lipids, which are primarily shown in green here, to carbohydrates and energy metabolism, as being familiar with the TCA cycle and glycolysis. And we could even connect that to amino acid metabolism and components of DNA. And so metabolomics cannot get all of these metabolites, but it does get a very large number. And so that when we look at the effect of a disease or the effect of nutrition, we can see a variety of these different metabolites being changed and then better understand the effect of the disease or the nutrition. And what has enabled us this ability is mass spectrometry and NMR. These two specific types of instruments have enabled metabolomics to grow and capture a large number of metabolites in one sample. And at the Cal Poly Metabolomics Service Center, which is where I am director, we do focus on this broad scale quantitation of metabolites in the body. We also look at nutrients in foods, and we use specifically ultra performance liquid chromatography mass spectrometry. And I have listed all of the different different assays that we have, and I'll be mentioning these a lot throughout this talk. So I just wanted to give an idea of when I say we're looking at these specific assays, what types of metabolites are we focused on? And so our primary metabolomics, we are looking at these types of ones, many of which are carbohydrates or energetic pathway intermediates. Our biogenic amines assays is getting amino acids, neurotransmitters, and we also have a lipidomics assay that gets many of these things such as phospholipids, lysophospholipids, sphingomyelins. And then we have 3 other assays such as bile acids, ceramides, and short chain fatty acids. And I wanted to give a little background on some of these specific metabolites because I talk about them very often throughout the rest of these slides. And cell membrane lipids and lipid mediators happen to be some of the metabolites that were most commonly altered in these vitamin A studies. So I just want to give a little bit of information about them. So lipid mediators they're also called bioactive lipids. They come from the cell membranes, and they are signaling compounds that you could find throughout the body, and they respond to different stimuli. And some of these are involved in inflammation, immune function, vascular tone. So they have a very critical role in the body. And some of the ones that I specifically be talking about are the lysophospholipids, sphingolipids, oxylipins, endocannabinoids, and bile acids. So see, these are some of the lipid mediators we talk a lot about. As far as the cell membrane lipids go, these ones primarily being phospholipids and sphingomyelins are part of that lipid bilayer in the cell membrane. And if we look at them more closely, they consist of phosphate, some sort of glycerol
de
back,
los que ya estamos hablando.
and 2 fatty acids. And so we'll be talking much about these cell membrane lipids. And the ones specifically are these phospholipids and sphingomyelin. And as you can see, these components of the plasma membrane have a lot of important function. They could be involved in inflammation, apoptosis. They do, of course, provide structure to the cell membrane, but they also are capable of releasing these lipid mediators. And some of the lipid mediators that they can release
Los que son
by
específicamente
specific
estos
enzymes,
fosfolípidos,
such as
y como ustedes pueden
PLA2,
ver en la
are
esfingomelina, y estas
lysophospholipids
son
and
las membranas que
lysophosphatidic
tienen
acid.
muchísimas funciones importantes y pueden
And
estar
these are involved
involucradas
in the cell
la
cycle,
inflamación, en la
inflammation, and immune
apoptosis,
response. Also,
y
ceramides
también para
have many of these similar functions.
ver estos intermediarios. Algunos
So some specific lipid mediators that we'll talk about a lot are the oxylipins, and these are found throughout the body, and they exhibit a really complex control over different systems in the body. And they're actually formed from the oxidation of polyunsaturated fatty acids. So if we kind of go back to that same picture with the phospholipids, another thing that they can release are polyunsaturated fatty acids. And when they're acted on by enzymes, they form oxylipins. So some of these that you may be familiar with are the omega-3 fatty acids and the omega-6 fatty acids. And from those, they are released from the plasma membrane, and then specific enzymes can convert them into a large number of other metabolites that have different.
y cuando están actuando,
and again, some of these main things that they control are inflammation, immunity, and things such as vasoconstriction and vasodilation. So the thing that really started this was that we did a study actually in a small animal model, specifically gerbils, and we just wanted to compare their liver metabolome in vitamin A sufficient versus marginally deficient gerbils. We ended up running 7 different metabolomics assays that are listed here. And very briefly, the altered metabolites primarily included sphingomyelids, phospholipids, lysophospholipids, which we had just mentioned previously on those other slides, bile acids, and acylcarnitines. And so that made us think that there might be some other avenues to investigate, especially concerning those in humans that might be changed. So there was a study that was performed by some researchers at UC Davis. They were doing a project in the Philippines, and we were able to acquire some samples from them and then run some metabolomics analysis. So I'll give a little more detail about that. So this was a pilot study, and we wanted to use metabolomics to look at differences in plasma associated with low vitamin A status versus those people that had adequate vitamin A status in lactating Filipino women. And we wanted to just understand the effect of vitamin A deficiency on metabolism in humans this time, though, because the previous study was in a small animal model. And then we also wanted to potentially uncover some biomarkers of vitamin A status. And so this small survey by UC Davis looked at the prevalence of vitamin A deficiency amongst lactating women. It was led by Doctors Engelstone and Haskell. And they saw that about 24% of the women had plasma retinol that was less than the optimal amount. And then 3.7% actually had deficient levels, which were less than 0.7. We were able to acquire a subset of those plasma samples and compare people with less than 0.7 micromoles per liter of plasma retinol, so that's deficient, versus those that were adequate that had higher than 1.05. We did metabolomics analysis, and specifically we looked at these 3 lipid mediators, and then we did these more broad analysis. And so this was 2 different labs performing this. There was Dr. Newman's lab at the USDA Western Human Nutrition Research Center doing this analysis, and then it was my lab doing this analysis. And these are the instruments that we used. And some background on the individuals in this study. There was a difference pretty much only in plasma retinol. So
tenemos estos análisis más amplios.
the vitamin A
Entonces
group were
tenemos
less than
estos
0.7,
distintos
and
en la
then the
USA
vitamin
y
A
también en el
sufficient
centro de
group was well over the
investigación
1.05
que fue
marker. And there were no differences in dietary intake. So the vitamin A content of their diet was similar. And when we looked at this, there was 28 of the 200 plus metabolites were significantly different, and 11 were these lipidomics metabolites, so mostly phospholipids and sphingomyelins. And so again, these are part of that plasma membrane. And so we could picture them as part of that plasma membrane here. They're actually formed in the endoplasmic reticulum, specifically these phospholipids. So I just have a pathway here. This is taken from one of our published articles. And so they're formed there. And then in the Golgi apparatus, that's where these sphingomyelins and ceramides are formed. And the down arrows just signify that all of these specific metabolites were decreased with vitamin A deficiency. And then also there were 13 oxylipins that were decreased with vitamin A deficiency. And they were mostly these LOX or lipoxygenase metabolites. 4 of them passed the multiple comparisons test. And so you see many of which were taken from arachidonic acid, that's omega-6 fatty acid. And many also were from this omega-3 fatty acid, EPA. So this pilot study, it gave us some more understanding of the impact of vitamin A deficiency on metabolic processes. And we were able to find some newer potential biomarkers. And many of these support findings of the gerbil study in terms of the sphingomyelins, lysophospholipid, and phospholipids, which was really interesting to see. But then also there are many newer findings that we found in these humans, such as the lower plasma oxylipins and endocannabinoids. And so those oxylipins, endocannabinoids are also lipid mediators. They are also having that control over things such as inflammation, immunity, and vasoconstriction. Now, the next study we did was with the University of Wisconsin and also with researchers in Zambia. And we did this one a little bit differently because, as was mentioned in the previous study, the focus was on whether the participants had a serum retinol that was below a certain level that was made them deficient versus those that had adequate vitamin A status. Now, the problem is that serum retinol as a measure of vitamin A status has many limitations and oftentimes can give an incorrect value based on certain factors. So in this specific study here in Zambia, they used what is considered the best way of measuring vitamin A status, and that is to look at the retinol isotope dilution test, which I'll talk about right now. And this is intended to give us the best idea of metabolomic changes in response to vitamin A status. So what are the problems with serum retinol? Well, although it is the most common biomarker for vitamin A status, it is often not accurate. One, because vitamin A is homeostatically regulated, so it's very tightly controlled in the blood, ranging from lower stores of vitamin A in your body to higher. It'll maintain a very similar serum retinol. It won't be until you have very low levels in the body or very high that it will actually change. Also, it's altered in the presence of inflammation and infection. So if there is the presence of inflammation and infection, the effect of that is often to decrease vitamin A levels, because vitamin A is carried in the blood by a protein that decreases with inflammation and infection. So essentially, it will give an incorrect diagnosis of vitamin A deficiency if those things are present. So the best way to assess vitamin A status is to use the retinol isotope dilution. And so what they use is this labeled retinol acetate tracer. So it's this form of vitamin A that they add an isotope label to it so that they could track it throughout the body and differentiate it from the vitamin A that's already in your body. And also they use gas chromatography, mass spectrometry to measure that in the blood. So what they're able to actually tell you is the total liver reserves, which is important because about 90% of the vitamin A in our body is in the liver. So if we are able to estimate liver reserves, that's a very good indicator of our actual vitamin A status. Furthermore, we could even estimate total body stores of retinol. And the way this works is that an individual will give a fasting blood sample. Then they will consume this retinol acetate tracer mentioned here. It will remain in their system, equilibrate for about 2 weeks. Then they will collect the blood and analyze the sample. And through some mathematical modeling, they are able to estimate the total liver reserves based on these 2 blood collections. So that is the best way to measure vitamin A status. However, it's very labor intensive and costly, which are the 2 main drawbacks of it. So the objective of this next study was to investigate whether with metabolomics, can we find metabolites different between Zambian women that had adequate versus deficient total vitamin A liver reserves in their bodies, in spite of them having no difference in serum retinol. So the serum retinol, that main way that we assess vitamin A status, they were not different in that, but it was obvious from their liver reserves estimated using the isotope tracing that there was a difference in their vitamin A status. So the study was taken in the Rufunsa district in the eastern part of Zambia. And this was a cross-sectional study. So again, they were just assessing the status of vitamin A amongst the people in that region, very similar to what they did in the Philippines study I just mentioned. And we took a very small subset of these individuals. Specifically, we took 10 people that had vitamin A deficient levels and compared them with 10 people with vitamin A sufficient levels. And for this one, we did some metabolomics analysis again, and these 4 assays were the ones that we focused on. And so you see here, these are the subject characteristics. And if we look at the baseline serum retinol, there is no difference between them. So the p-value is 0.66. And so if you were to look at these individual groups of people based on the most commonly used vitamin A assessment measure, which is serum retinol, these people would not only be, they would be both sufficient in vitamin A and they would not be different in vitamin A between the two groups. But when we do the retinol isotope dilution test, these people, so those that were vitamin A deficient had a much lower amount of this total liver reserves in total body stores than did those that were vitamin A sufficient, with a highly significant p-value in terms of that difference. And for reference, total liver reserves less than 0.1 are considered vitamin A deficient by bond. And then there's also another specific way of measuring that they believe it should be less than 0.07. But in any case, the mean level in the vitamin A deficient people was 0.06. So it was below both cutoffs for vitamin A deficiency. And when we looked at these metabolites and the differences between the groups, we did see similar changes as we did with the Filipino women. And in the specific case that, again, it was these plasma membrane lipids that were different between the groups, as well as the lipid mediators. And so if we kind of zero in again on those cell membrane lipids, we saw lower phosphatidylethanolamines and phosphatidylcholines. So the specific phospholipids were lower. We also saw lower choline, which is derived from phosphatidylcholine. And we saw that cholesterol esters were higher. So that was a little bit newer of a finding. And oxylipins were lower again. So those lipid mediators that have control over these different functions, such as immunity and vasoconstriction, vasodilation, and inflammation, they were again down. And so now we've seen this pattern in both the samples taken from the Philippines and those taken from Zambia. And we're starting to see that there is this consistent effect with vitamin A deficiency on these markers. And understanding these effects is a bit difficult because these are relatively new discoveries. There is some research out there that shows that vitamin A deficiency does impact the synthesis of these phospholipids. The exact mechanism for that is not quite known, but there is some evidence that they get impacted. And for example, some of the specific derivatives that are needed to produce these phospholipids, there's actions upon them that gets changed. So we really, though, still need to figure out why exactly these phospholipids are lower with vitamin A deficiency. So this is definitely something that would be good to look into in the future and understand that better. Also, interestingly, with these oxylipins that are involved in these different functions, most often with disease processes, oxylipins are actually higher. You see them higher in in things such as diabetes and cancer. Like a lot of times the omega-6 forms are higher, but we just see like a completely across the board decrease in these oxylipins, which is a bit unusual. And what it suggests to us, though, is that there is just perhaps a less ability to regulate these specific processes. And this may also be related to the decrease in the phospholipids, because these oxylipins are derived from them. However, we do need more evidence to suggest whether these enzymes are being affected that produce the oxylipins, or is it simply a matter of there being less phospholipids, so therefore there's less oxylipins. So there's a lot more to be discovered and understood regarding these changes, but we are seeing a consistent pattern, which is very interesting to us, and we'd like to understand it more. And these are avenues that really haven't been explored much in the literature. Now, so that's kind of the aspect in terms of the metabolic effects of vitamin A deficiency and what it might be doing. The other side of this is that we would potentially like to use these as biomarkers of vitamin A status, because as I was mentioning, that retinol isotope dilution technique, it is a very good technique, but it is labor intensive. It costs a lot of money. It's not practical using on a large population. And for assessing vitamin A deficiency, we really need to be able to assess large populations. And using the retinol isotope dilution as it stands is not something that's practical. So we are trying to find other biomarkers that could be used in their place. And so when we did this correlational analysis, which I want to very much state that this is a small sample size and we cannot take that much from it because of that small sample size. But just to note some interesting observations was that correlating with the total liver reserves were some phosphatidylcholines, lysophospholipids, and some other lipid mediators did have some moderate correlations with total liver reserves. Of course, we need much more research on this on a larger population, see if this maintains this correlation. Also across a range of vitamin A levels, like right now, we're just talking about vitamin A deficiency versus sufficiency. We need to see also what might be a better marker for subtoxic or toxic vitamin A concentrations. But just to get us starting to thinking about biomarkers, and since this is new research, we're starting with a small sample size, and we hope to increase that in the future. So select phospholipids and oxylipins were capable of differentiating the Zambian women with adequate versus deficient total liver reserves, despite them not having differences in serum retinol. And so future studies of a larger sample size with a wider range of total liver reserves are really needed to determine if these are reliable biomarkers for assessing vitamin A status. Now, I do want to kind of switch gears here because I was just focusing on those previous studies on vitamin A deficiency, and that has been a big focus over many, many years due to its high prevalence in different areas of the world. But we also want to kind of look at the toxic range of vitamin A and some important aspects to consider regarding that. And so in order to respond to the high amounts of vitamin A deficiency in certain regions of the world, there have been a lot of different interventions used. So we've used supplementation of vitamin A, fortification, micronutrient powders, diet diversity, biofortification. So we've attempted many different strategies to address vitamin A deficiency. And over the years, it certainly has had a positive effect in terms of reducing the amount of deficiency in certain populations. And so we see that in some of these assessments, we see that Southeast and East Asia, we've seen a reduction of vitamin A deficiency prevalence from 42 to 6%, Latin America and the Caribbean from 21 to 11%. Sub-Saharan Africa has not changed as much as would have been hoped, and South Asia the same. But there are some specific regions where it has had a dramatic effect, and others maybe not as much, but there are other factors involved that might be influencing the ability to accurately assess the prevalence. But this may be linked to the reduction in 5 years of age mortality rates, as well as there has been a reduction in that of nearly 50% since 1990, and reduced deaths due to measles, diarrhea, and malaria, that it is believed that vitamin A supplementation has had a large impact on those positive outcomes. Now, one thing that we're beginning to look at more is vitamin A toxicity prevalence. And that's because of potential overlapping vitamin A deficiency, overlapping vitamin A interventions in different regions of the world. And in fact, there has been some evidence of toxicity in the USA, Zambia, Guatemala, and South Africa. So in Zambia, they did a study a little bit ago, and when they surveyed prior to the beginning of the study using serum retinol as the main measurement, they found that there was a fair amount of vitamin A deficiency in this region. And we have mentioned previously kind of some of those limitations of serum retinol and things such as infection and inflammation have a potential effect on them. But in this study specifically, they used the retinol isotope dilution technique. So they were able to estimate total liver reserves, which is seen as the gold standard and is the best way to measure vitamin A status. And so when they performed feeding intervention in that study that had been stated to have vitamin A deficiency, and they conducted the study and ran the analysis and came back and found that based on total liver reserves, 59% of the children had hypervitaminosis A. So 59% of the children in that study actually had very high levels of vitamin A. So this is much contrary to what they had suspected, was that they suspected that the region had a good amount of deficiency. So the question then was, how could have this happened? And it began to be considered that perhaps there were a lot of different interventions occurring that led to the levels of vitamin A in these children to be very high. So for one thing, there is high dose supplementation for for 6 months old children to less than 5 years old. So during that time period, these children receive high dose vitamin A supplementation. Also, there has been an increase in fortification in that region. And so there's been some mandated, so required sugar fortification using retinol palmitate. Also, they've begun biofortification. And specifically, they've released what's called orange maize, which is a corn that has been crossbred to have higher levels of beta carotene, and beta carotene thus is a source of vitamin A in the body. Also, orange flesh sweet potatoes in a similar way have higher amounts of beta carotene and thus are a really good source of vitamin A. Then there was a promotion of diet diversity. So the Ministry of Agriculture promoted red palm oil intake. And so some regions had diets considerable also of vegetable and fruits high in provitamin A carotenoids. So as they promoted consuming foods that had higher amounts of vitamin A, this also contributed to the amounts they were consuming per day. And so there was kind of a combination of different interventions that likely led to these very high levels of vitamin A in this region. And so now the thought shifted to that we need biomarkers for toxicity as well to properly screen populations prior to intervention. So prior to going in and promoting these interventions or instituting these interventions, such as supplementation, fortification, biofortification, and diet diversity, we need to measure the blood and definitively say whether the population has the vitamin A deficiency, or in some cases may even have vitamin A toxicity. And so finding biomarkers for that has now become a focus. So that brings us to the last study I'll talk about, which was looking at vitamin A supplementation in South African preschool children and the potential effects that we were going to analyze using metabolomics. This was a study, again, with the University of Wisconsin, who are experts in that total liver reserves technique. And then also this is with the South African Medical Research Council. And so we really wanted to assess the metabolomic changes occurring after supplementation of vitamin A. And so the way that this was done was that we took a subset, a small set of serum samples from children that had either adequate total liver reserves of 0.59 or had toxic vitamin A status, so about 1.7 micromoles per gram. So there were a fair amount of children that had this range of total liver reserves that would be considered toxic and toxic vitamin A concentrations. So we looked at samples both before and 1 month after they consumed a 200,000 IU vitamin A capsule. So this is part of that supplementation program I mentioned that's seeking to increase the levels of vitamin A and in essence, decrease vitamin A deficiency. This is looking specifically at those children before and after consuming that supplement. And by this time, we know that the changes being observed in due to vitamin A status are largely related to the compounds we see with our lipidomics and oxylipin assays. So those are those cell membrane lipids and lipid mediators that are largely being changed. So for this analysis, we only looked at those two. And when we looked at those, we looked at it in two different ways. So we looked at it in terms of adequate versus toxic vitamin A status, as well as pre and post supplementation. So this initial analysis shown here, we're focused on looking at the adequate versus toxic vitamin A status. And as you can see here, comparing the adequate vitamin A before supplementation to the toxic vitamin A before supplementation, there were 13 phospholipids and lysophospholipids that were lower in the toxic group. So with that really high level of vitamin A, they had lower amounts of these specific metabolites. And then we also looked at after supplementation. So if we compare the adequate vitamin A status group after and to the toxic vitamin A status after, many of those same phospholipids and lysophospholipids were higher in the toxic groups. So there seemed to be some sort of effect from the supplementation that was reversing the differences between groups. And so to follow up on that, we then ran a mixed model accounting for specific covariates and focused more on the effect of group and time. So when we looked, there was 22 metabolites that were altered post-supplementation. So this was the effect of consuming the supplement over time. So from the before supplement to after supplement of vitamin A, we saw elevated phospholipids and lysophospholipids in general, and then we saw decreased oxylipins. And then if we consider both the group, so in other words, adequate versus toxic vitamin A status groups, with the time, we saw that 28 metabolites were altered, including phospholipids and lysophospholipids, and they were elevated in the toxic status group. So this line graph shows that with high vitamin A status before supplement to after supplement had this dramatic increase of this lysophospholipid, which is really meant to be a representative lysophospholipid because many of them had this similar kind of change. And then low vitamin A status was more or less unchanged. So there was definitely a difference in response to the vitamin A supplement based on whether you consumed it and you had adequate vitamin A already, or you already had toxic vitamin A. And so what will be interesting to look into the future is why exactly that's happening and what potential effects that has when you have this increase in these phospholipids and lysophospholipids. So these types of things definitely need investigation. And then also we really need to look into more studies like this with a wider range of vitamin A status so that we could see if some of these could be potential biomarkers. Now, because again, this was another small sample size study in a specific region and in children. So more research is definitely needed to draw major conclusions, but this does help contribute to what we're learning. And the last study I want to talk about is something that is very new data. Haven't really been able to get far into data analysis and potentially seeing how it could be complementary to some of the other studies. But I just wanted to show some very simple results, simplistic analysis that we've conducted so far. And so this was with vitamin A total liver reserve associations with biomarkers in women from the United States. And so this was a pilot study, and it was just taking samples from an intervention study that had been conducted in the United States. We took the baseline samples from 10 women, and we looked at how the total liver reserves correlated with different metabolites. There were some moderate correlations that were observed with phosphatidylethanolamines, positive correlations, and ceramides, negative correlations. But again, we need to do a lot more analysis and looking more into this, but I just wanted to show some kind of results for today and how, now we are applying this in the USA, where the USA also has had some suggestions of issues with a high prevalence of toxicity of vitamin A due to the large amounts that are consumed there. But there's still a lot left to be known about these size, that these are all small sample size studies. These are just kind of the beginning of the investigation. So what we really need to look into are large populations with a wide range of vitamin A status, deficiency, adequacy, toxicity. We need multiple collections per individual. We need to look at different regions of the world to see how things, if these things can be translated to different areas. Varieties of age, sex, gender, and all of this needs to have the retinol isotope dilution data, because that's what's really giving us the accurate vitamin A status. Furthermore, we need to look at infection and inflammatory biomarkers, because we know that those can affect vitamin A levels, and also take detailed dietary records, because with these metabolites that we're investigating, we also need to rule out or try to rule out diet effects on them. So they need to be able to be markers that aren't changed depending on your diet intake of things other than vitamin A. So I just want to acknowledge some of the funders as well as some of the people that support our research, and some of our current lab members as well, such as technician Rob Fancher and many of the great students that I work with. Thank you.
Thank you, Michael. It was a great talk. We learned about many things. For example, the effect of vitamin A in phospholipids in membrane, in cell membrane. That thing, this is a huge finding. Let me see if there is some questions from the audience.
Okay.
Yeah, there is one. Dr. Olga Garcia Obregon say, excellent talk. We have done studies looking at vitamin A status in blood in obese women from rural areas of Mexico. Your research has been done in population with deficiencies and probably undernourished. Do you believe that the metabolites that you find in these populations will be also present in Mexican women with obesity and vitamin A deficiency?
Yeah, so that's an excellent question, and definitely something that is going to require us to look at also the impact of weight, or looking at associations with BMI to really understand if that might be a factor. I could only say that with the analysis that we've done, we've used BMI as a covariate to try to, in our statistical analysis, in order to try to reduce the influence of BMI on our statistical results. However, because we are looking at a small sample size, I'm hesitant to say that rules out BMI as a potential factor. So I would say that that future research, we're going to need to get more information with people with wider ranges of BMI to truly understand if these biomarkers would still be reliable in spite of weights. So excellent.
Okay. Yeah. Thank you, Michael. We are short in time right now, but we appreciate a lot your participation in this meeting, and we hope to have you here and have great collaboration. Dr. Olga Garcia Obregon is also in our group of research. Then I think that we are going to be starting a great collaboration with you. Thank you very much.
Thank you. Thank you so much. Thank you for the invitation. Have a good day.
Bye-bye.
Bye. Bye.
Hola.